So my name is Mike Collis. I'm the Chief Executive of the Physiological Society, and I'm here in Manchester at our main meeting. The Physiological Society is a very august and important society. It started in 1876, and every year we have a major meeting where research is presented from our members and from scientists all around the world. About 1,000 people at this meeting today. What I'm going to do soon is to introduce the GSK prize lecturer, Josef Kittler. And this prize lecture, which is supported by GSK, we're very pleased they support it and hope they'll continue to do this. It's meant for young scientists the society considers are rising stars, and it gives them the opportunity to present their work to a much broader audience at a meeting like this, where there may be over 1,000 physiologists. And Dr. Kittler's work is about inhibitory synapses in the brain and how they're regulated. It's important to both health and disease, and he uses a range of very novel techniques, patch clamping of neurons, and also imaging techniques which use nanoparticles to show organelles within the neuron moving around. His background is he trained at UCL in the MRC cell biology unit, and he stayed at UCL. He's had a period of collaborating with David Atwell, who's a well-known and very eminent member of the society. And he now holds an MRC Career Development Award there.
Thank you very much for that very kind introduction. It's a great pleasure and an honour to be giving the opening lecture for PhysOc 2010. And today I want to tell you about some of our recent experiments where we're trying to better understand the molecular mechanisms that are important for regulating the strength and plasticity of inhibitory synapses in the brain. So, as I'm sure you're all very well aware, compared to many other cell types in the organism, neurons have a particularly complicated cellular and subcellular architecture. You can see this from these very beautiful drawings of pyramidal neurons in the cortex by Ramon y Cajal. So they have an axon that can extend many thousands of microns, an elaborately branched dendritic arborization onto which impinge many thousands of synapses, which mediate the communication between the different neurons in the brain. And because of this complicated architecture, they need a specialized mechanism to localize, maintain, and remove proteins and also organelles from discrete locations within this complicated cell. And also they need to be able to tailor this to different levels of neuronal activity. So in my lab we're interested in how these mechanisms, important for discretely localizing proteins and organelles within the neuron, may contribute to neuronal development and also the formation and plasticity of synaptic connections. So there are two main types of synapse in the central nervous system, as I'm sure you're well aware. Excitatory synapses, where glutamate is released to activate postsynaptic ionotropic glutamate receptors like AMP receptors and NMDA receptors, and this allows a sodium and calcium influx into the cell, causing neuronal depolarization and an increased likelihood of action potential firing. And inhibitory synapses, where GABA is released, which allows a chloride influx into the cell through postsynaptic GABAA receptors, which leads to hyperpolarization and a decreased likelihood of action potential firing. And it's these inhibitory synapses that we're interested in. So they play a particularly important role because, so here we have a presynaptic excitatory cell, releases glutamate. If enough synapses get activated, there'll be a sufficient depolarization to start that cell firing, whereas the inhibitory synapses play a crucial role in mediating general inhibition, stopping cells from becoming too excitable, which could lead to runaway excitation. But also, by generating temporally and spatially precise inhibitory synaptic input, they can help to fine-tune a neuron's responsiveness to excitatory input and therefore temporarily regulate its activity. And that has important implications for generating discrete activity patterns in the brain. So the strength of inhibitory synapses is going to play an important role in shaping the input-output relationship of neurons, and that can have profound effects in terms of information processing in the nervous system. So the workhorse postsynaptically of the inhibitory synapse is the GABAA receptor. It's a member of the ligand-gated ion channel superfamily, and it assembles from 5 subunits to line a chloride-permeable pore. There's a large subunit heterogeneity, but it's generally agreed that synaptic GABA receptors tend to comprise of 2 alpha subunits, 2 beta subunits, and a gamma 2 subunit. Now, the N-terminus of this receptor contains interaction domains for mediating receptor assembly, and it's also the locus for making agonist and allosteric modulator binding pockets. But from our perspective, it's also useful because it allows us to engineer sites within this N-terminus such that allows us to sort of facilitate the imaging of these receptors at the plasma membrane. Each subunit comprises 4 transmembrane domains, and there's a large intracellular loop between TMs 3 and 4. And this is a site for post-translational modifications of the receptor, which is an important mechanism whereby the receptor can be regulated by intracellular signaling pathways within the cell. In addition, this intracellular domain mediates protein-protein interactions with proteins in the cytosol that are important for regulating various aspects of the biology and also the membrane trafficking of the receptor. And so I'm going to focus on how post-translational modifications and also protein interactions are important for regulating the biology of this receptor and also the strength of inhibition in the brain. So quite a lot is known about the components of this inhibitory synapse. In addition to the postsynaptic GABA receptors, there is also some adhesion molecules that are important for ensuring the correct apposition of the presynaptic terminal opposed to these postsynaptic receptors. In part, that's mediated by a transsynaptic adhesion complex that's composed of presynaptic neurexins and the postsynaptic neuroligin molecule. And GABA receptors and neuroligands are in intimate association with an inhibitory postsynaptic scaffold molecule called geferin, which is thought to form a proteinaceous lattice underneath the plasma membrane that's important for confining the GABA receptors within this postsynaptic domain. But this sort of gives the impression then that this synapse is quite static in terms of the GABA receptors. But in fact, work from a number of groups, including work that I carried out while I was working with Steve Moss at UCL, we actually found that the GABA receptors are highly dynamic in the plasma membrane. So receptors at the cell surface, for example, can exchange between synaptic and extrasynaptic compartments. We also identified a mechanism whereby receptors can be down-modulated from the plasma membrane via clathrin-mediated internalization. The receptors are stored from this down-modulation in an internal storage compartment, and from there they can either be targeted for degradation in the lysosome or rapidly recycled back to the plasma membrane. So there's a constant cycling of receptors between surface and intracellular compartments, and also between the synapse and extrasynaptic regions. And what I hope to convince you of today is that one very simple and effective way to regulate the strength of this synapse is by changing basically the kinetics of some of these trafficking pathways and simply regulating the number of the postsynaptic receptors. So to give you one example of some work from another group which showed really very nicely in a physiological context how this inhibition might be important, I just want to mention some work from Toru Kurantani and colleagues. So they were interested in studying the mechanisms of plasticity of somatic inhibition in the cortex, and they were looking at layer 5 pyramidal cells within the rodent cortex. And what they found was that when they looked at neurons that were from an awake animal, there was significantly reduced inhibition compared to the levels of inhibition that occurred in an animal undergoing slow-wave sleep. And they were interested to understand what the mechanisms of this plasticity of inhibition was underlying this change in the strength of the synapses. And what they found was that if they mimicked firing patterns typical of awake animals, this produced this long-lasting depression, which was due to the activation of L-type calcium channels driving the endocytosis of GABA receptors into this internal compartment. Whereas in contrast, firing patterns that were similar to those that one sees in slow-wave sleep actually produced a long-lasting potentiation dependent on the activation of a different type of calcium channel, which drove the exocytosis of receptors. So this suggests then that rapid bidirectional modification of inhibition dependent on GABA-A receptor trafficking and basically the movement of receptors between different compartments can actually have a profound impact on cortical network activity during different behavioral states. Okay, so now I kind of want to zoom back in onto our focus on really the molecular mechanisms, and my talk is going to be broken down into 3 parts. So in the first part, I want to tell you about the mechanisms that are important for regulating one mechanism that's important for regulating the lateral diffusion of receptors. So it's been known for a long time, for a while at least, that the receptors can exchange between these 2 compartments, but the underlying machinery that might regulate that process, and whether, for example, it can be regulated by changes in neuronal activity, have remained unclear. So after that, I want to talk about what happens to the receptors after they've been downmodulated from the cell surface via this endocytosis process. So receptors are moved to this internal compartment, and as I said, they can either be targeted for degradation or they can be rapidly recycled. And we're interested to know what determines that. Is there a molecular switch that might, for example, facilitate the sorting of receptors into this degradative pathway? And in my second part of my talk, I want to focus on this region. And then finally, while this internal store of receptors is very important for replenishing the number of postsynaptic receptors at the synapse, the machinery that mediates that delivery of receptors from inside the cell has remained elusive for quite a long time. And in the third part of my talk, I hope to tell you about some of the mechanisms that are important for that process. So, to start off, I want to tell you about some work carried out by a talented young student in my lab called James Muir, who joined the lab really with the intent of trying to be able to follow the membrane trafficking and surface stability of these GABA receptors at inhibitory synapses. And to do this, he has established 2 different types of imaging approach so that we could actually visualize in real time in neurons with high temporal and spatial resolution the behavior of the receptors. So the first way that he did this was to take advantage of a variant of green fluorescent protein that Gero Miesenbock had, while working with Jim Rothman, had engineered to make much more pH sensitive. And what that means is that when we introduce this GFP into the N-terminus of the GABA receptor, so we engineer a GABA receptor subunit with an N-terminal tag with this pH-sensitive GFP, when it's at the cell surface and we're imaging in an imaging media containing with a neutral pH, this protein is highly fluorescent and very bright. On the other hand, when we're inside the cell, so when the receptors are localized in internal membrane compartments, the GFP, this pH-sensitive GFP, is pointing into the lumen of these internal compartments, which is slightly acidic, and that causes an eclipsing of the fluorescence. And so that basically gives us a nice way to be able to only visualize the surface receptor population. And we can transfect into neurons, for example, this cultured hippocampal neuron here, a GABA receptor subunit with this tag. What we know is that this subunit assembles with the native receptor population and is effectively targeted out to clusters of GABA receptor accumulations out in the neuronal dendrites. And I won't show you the data, but we know that these are opposed to inhibitory presynaptic terminals. So we have a readout now for being able to look at the stability of these clustered GABA receptors at inhibitory synapses in real time using imaging approaches. So this is an example movie that James has made using live cell video microscopy of these GABA receptor clusters opposed to synapses out in neuronal dendrites. And what we found was that we can image for minutes, tens of minutes, hours. It doesn't make much difference. The receptor clusters are extremely stable under resting conditions. We then wanted to know whether or not changes in neuronal activity, for example, by driving the activation of glutamate receptors at excitatory synapses, could have any influence on the stability of these postsynaptic GABA receptors. So is there any crosstalk between the activation of glutamatergic transmission and synaptic inhibition, which might have important implications in terms of information processing? So, to look at this, James carried out imaging experiments where, the movie's on a loop here, where he imaged the fluorescence in these clusters during and then after the activation of glutamatergic signaling by perfusing on glutamate. And what he found was that there was a rapid and significant loss of the postsynaptic GABA receptor fluorescence within a few minutes of activating these excitatory synapses. So I hope you can see that basically in the movie. So this suggests then that activity might in some way influence the stability of the receptors. So we can quantify this change by drawing regions of interest around each individual receptor cluster. And under resting conditions, as I told you already, the receptors are very stable. There's very little loss of fluorescence. There's essentially no photobleaching. But if, on the other hand, we activate glutamatergic signaling by perfusing on glutamate, then we see this rapid within a few minutes down-modulation of the intensity of these clusters. And it's somewhere in the region of 35 to 40% of the receptors that are down-modulated during this time. So what's the mechanism? Well, what we found was that it was blocked by the NMDA receptor antagonist APV, and it was also, this down modulation was blocked in the absence of extracellular calcium in the recording solution, suggesting then that this is mediated via calciumentry through the activation of NMDA receptors. It's not actually dependent on the activation of the GABA receptors themselves, because we still see the down modulation in the presence of a GABA receptor antagonist bicuculin. So this suggests then that we have a mechanism whereby the activation of glutamatergic signaling can influence the stability of GABA receptors at inhibitory synapses. Now, the way we did those experiments was to globally activate glutamatergic signaling, but more realistically, what would be likely to happen is just that a few synapses might get activated at any one given time. So rather than all the excitatory synaptic transmission being activated, it's much more likely maybe that just a few synapses could be driving this declustering. So we were interested to know whether we could see a localized declustering in the vicinity of just a small number of excitatory synapses being stimulated. So to look at this, James teamed up with Andrew McCaskill in the lab, and they used the same imaging approach. So again, imaging the clustered GABA receptor fluorescence out in the neuronal dendrites, but then they morphologically identified some fine axonal processes crossing this dendrite, and then used a small micropipette to stimulate locally the axons here, and then looked at whether or not there was any difference in receptor declustering here versus further away. And they found that actually they saw a localized declustering just in the region in proximity to where we have some presumed synaptic contacts that are being activated. And so this suggests that actually even a local activation, a local stimulation of a few excitatory synapses might be sufficient to then locally disperse GABA receptors and reduce inhibition in a small region of a neuronal dendrite. So we also wanted to know a bit more about the mechanism that's driving this dispersal of GABA receptors from these inhibitory postsynaptic domains. When James looked at the total change in fluorescence in the dendrites, rather than by drawing regions of interest around each cluster, there was no change in total surface receptor fluorescence. So that suggests then that it's not a downmodulation of the receptors from the plasma membrane, but rather that the receptors are simply moving out of the synapse by, for example, lateral diffusion. And we wanted to be able to test that in a different way. And so one of the caveats of using the Super Ecliptic Phluorin approach is that it's not sensitive enough to look at the behavior of single receptors. The GFP is just simply not bright enough. It's good for looking at receptor aggregates below the synapse, so probably aggregates of 25 to 50 receptors, but it doesn't really tell you anything about the behavior of single receptors within those neuronal subdomains. So James took advantage of a new set of fluorophores, some kind of new kids on the block, so to speak, of fluorophores available for carrying out biological experiments called semiconductor nanocrystals or quantum dots. And these are, as the name might imply, very, very small and also very, very bright fluorescent molecules. You can just think of them as any other fluorophore. And they have the advantage that they're so bright that with a very sensitive CCD camera you can detect a single quantum dot, and also they're extremely photostable, so that you can image them for long periods of time. And so he took advantage of the fact that you can attach quantum dots to antibodies, and then used an antibody to GFP, and then targeted this tag that we'd engineered into the GABA receptor, this GFP tag. And that allowed us then, if we sparsely labeled the receptors in the dendrites, to be able to image simultaneously the large GABA receptor clusters or aggregates at synapses, and also a few of these individually labeled GABA receptors with quantum dots. And this is a movie, an example of the behavior of a single quantum dot, so the clusters are very stable, but individual receptors, in contrast, actually can move and are very dynamic. So here the receptors are moving presumably just by Brownian diffusion in this extrasynaptic compartment. It then enters at some point into the synapse. Something tells it that maybe it's good to hang out here because it spends about 30 seconds in this domain, but then eventually it gets bored and sort of leaves again. But the take home here is really that individual receptors are actually can be very dynamic, and they can rapidly exchange between synaptic and extrasynaptic compartments, and that might be an important mechanism for regulating the number of postsynaptic receptors. And using this sort of approach, it was then possible to look at whether or not there was a change in the mobility of single GABA receptors under conditions where we activated glutamatergic signaling. So this is the movement of single GABA receptors using this imaging approach in a neuronal dendrite under resting conditions. So there are still some that are quite dynamic and moving around. But this is very significantly increased when we activate glutamate receptors. So this is after perfusing on glutamate. And what I hope you can see is basically that the Q dot labels receptors have got a much more increased mobility. And we can get sort of useful parameters out of this, so we can reconstruct the tracks from these quantum dot labeled receptors under control conditions. The tracks are longer under glutamate when we activate glutamate receptors because the receptors are more mobile, so they explore more of the dendritic area. And then we can also look at the mobility of the receptors inside the synapse and also outside of the synapse. And what we find, for example, if we plot here in this cumulative distribution the diffusivities of receptors both inside and out of the synapse before and after the activation of glutamatergic signaling, we see that there's a shift to receptors that are more mobile. And this happens under both conditions, but it's much more the case actually at the inhibitory postsynaptic domain. So basically, activating glutamate receptors makes the GABA receptors less confined or more mobile at inhibitory synapses, and that actually correlates with a reduction after glutamate receptor activation in the residency time of individual GABA receptors at those synapses. And we think that's the mechanism that's basically underlying the loss of postsynaptic receptors. One advantage of the quantum dot approach is that one can also do this with native receptors, and basically we find that they explore more of the surface area, these native receptors, after activating glutamatergic signaling, as can be seen by the maximal projection of the surface area explored before and after here, and we also see this shift to higher mobilities when we calculate the diffusion coefficients of the individual receptors. So this suggests then that what might be going on here is that basically activating NMDA receptors leads to a calcium influx, which somehow makes the GABA receptors less stable at this postsynaptic domain. But we also wanted to know what the mechanism might be at the molecular level. So to look at this, we basically thought, well, it must be a calcium-dependent signaling pathway that's driving or changing the stability of these postsynaptic receptors. And to cut a long story short, we basically honed in on a calcium-dependent phosphatase called calcineurin. And so, as I showed you before, the receptors are pretty stable under resting conditions. When we activate glutamate receptors, we see that dispersal, and we block that in the presence of an inhibitory peptide that blocks the function of this calcium-dependent phosphatase that acts to dephosphorylate substrates. And we see the same thing with another calcineurin inhibitor, cyclosporin A. So this suggests then that in some way this phosphatase is dephosphorylating a substrate, and that somehow changes the stability of the receptors at the synapse. Now, there could have been a number of different substrates, but actually there was already some information in the literature that pointed to the fact that there was a phosphorylation site within the GABA receptor itself that was a known target for dephosphorylation by calcineurin. So as I mentioned at the start of the talk, this intracellular domain can be phosphorylated and dephosphorylated by a number of different kinases and protein phosphatases, and we were already aware that a specific serine site within the gamma-2 subunit of the GABA receptor, serine 327, was a known site dephosphorylated by calcineurin from work by Yuming Liu. So we wanted to know whether indeed that serine site within the intracellular domain might actually be mediating this activity-dependent redistribution of the receptors. So to be able to test this with mutant versions of the receptors, we took advantage of the Super Ecliptic Phluorin tagging approach again, because in addition to it allowing you to visualize receptors, one of the other advantages is that it can allow you to put in receptors into the system that have, for example, a particular point mutation. So we basically repeated the experiments that we'd done already, but this time using a gamma-2 subunit of the GABA receptor tagged with this super ecliptic Phluorin. We initially ensured that receptors containing this tagged subunit were again efficiently targeted out into neuronal dendrites. So what I hope you could see is these clusters of fluorescence again. So it's effectively targeted out into the right place. They're opposed to inhibitory presynaptic terminals, and they also undergo activity-dependent receptor declustering, which I hope you could see over time here. When we did the same, so here's just the plotted fluorescence change over time. Again, when we activate glutamatergic signaling, we lose that fluorescence. When we did the same experiment in the presence of the mutated version of the receptor that can no longer be dephosphorylated by calcineurin, we didn't see that effect. So basically, mutating the serine 327 site occludes the ability of the NMDAR receptor signaling to downmodulate or cause the driving out of GABA receptors from this inhibitory synapse. So we also wanted to know whether or not that might have any effect at the level of single GABA receptors. And so in this case, James took advantage again of the quantum dot tagging approach to label up some of these gamma-2 subunits tagged with super ecliptic Phluorin, and then looked at the mobilities of receptors. And again, what he found is that there was a much more significant shift to higher mobilities in tracked receptors that contain the wild-type subunit versus receptors containing the mutant. And then when we looked at how long individual receptors spend at the inhibitory postsynaptic domain in the case of the wild-type, again, this was significantly reduced when we activated NMDA receptors, but we didn't see that reduction in residency time at synapses containing this phosphorylation mutant of the receptor. So that suggests then that the mechanism by which these GABA receptors are changing their lateral mobility to lose this confinement at the inhibitory postsynaptic domain is going to be dependent on calcium entry through the NMDA receptor, leading to the activation of calcineurin, which translocates to the receptor and then causes the dispersal, probably not quite as fast as that, but causes the dispersal of these receptors from inhibitory synapses. And I think that this has some important and potentially interesting implications in terms of local regulation of the strength of synaptic inhibition mediated by changes in neuronal activity. So, for example, it suggests that if you activate one of these excitatory synapses, you could locally disinhibit the surrounding region. And one might imagine that, for example, that could facilitate the activation of other excitatory synapses within the vicinity, and maybe also facilitate the potentiation of those excitatory synapses during synaptic plasticity. This may also have important implications in terms of pathology as well, because if you get high levels of glutamatergic activity and lots of calcium going in, then that might cause a much more sustained loss of GABA receptors within the plasma membrane from their synaptic sites, and that might lead to disinhibition, which one could imagine could lead to the generation of too much activity, runaway excitation, and, for example, the generation of seizures. Okay, so in this first part of the talk, I talked about the stability of GABA receptors at the cell surface. But now what I want to do is focus on what happens to receptors once they've been downmodulated from the plasma membrane. So as I mentioned at the start of the talk, receptors are actually constantly cycling between an internal stall and the cell surface. And in work that I carried out a number of years ago, we were able to show that, in fact, if you interfere with some of these parts of the pathway, that can significantly affect the number of postsynaptic receptors. So this internal store of receptors is very important for being able to provide a pool of mobile receptors that can be rapidly returned back to the plasma membrane, potentially to change the strength of this inhibitory synapse. But we also know that the receptors can be targeted for degradation, and Lorena Arancibia-Carcamo work between my lab and the labs also of Steve Moss and Trevor Smart, and wanted to address what it is that might determine whether or not a receptor maintains a stable, is maintained stably here or is recycled, or rather can be targeted for degradation in this compartment called the lysosome. So before she set out to really figure out what the molecular mechanism of this trafficking might be, she initially wanted to ensure that actually, or at least find out whether or not changing degradation of receptors within this compartment could actually have any influence on the number of postsynaptic GABAA receptors. And the way she approached this was to use an inhibitor of lysosomal proteases called leupeptin. So it's possible to treat cells with leupeptin. That's going to block the function of the proteases within this compartment, and that leads to blockade of this degradative pathway. So using this sort of approach, she then went on to investigate whether or not blocking GABA receptor dependent degradation in the lysosomal pathway was in any way influencing the number of postsynaptic receptors. And the way she did this was to make acute cortical brain slices, rodent brain slices, in the same way that you would make slices, for example, for electrophysiological recordings. She maintained those slices in oxygenated ACSF, and then treated the slices with leupeptin, and then looked at whether or not there was a difference in the number of, or the size of GABA receptor clusters under controlled conditions versus blocking this pathway. So here we have one example of a neuron from one of these untreated or treated slices. The slices have been labeled with antibodies to the GABA receptor to be able to detect the postsynaptic GABA receptor clusters. They've been labeled with an inhibitory presynaptic marker so that we only look at the synaptic receptors, and also with a nuclear stain so that it's easier to find the cells within the slice. And here is just an intensity map, basically, for the extent of GABA receptor fluorescence under control conditions, or after we treated neurons for 3 hours with this inhibitor of the proteases, the lysosomal inhibitor. And what Lorena found was that there was a significant increase in the size of these GABA receptor clusters, which can be seen here in these cumulative distribution plots. So basically, the size of the inhibitory clusters is shifted to the right here, and so this is some of the pooled data. So basically the clusters get bigger, and we also found that there were more of these inhibitory postsynaptic clusters. So this supports the idea then that actually that degradative pathway under obviously slower timescales than the timescales that I was talking to you about in the first part of the talk. So rather than seconds to minutes, we're talking about hours here. But within a couple of hours, we can dramatically regulate the number of postsynaptic receptors via this targeting of the receptors to the lysosome. In agreement with that, we looked at whether or not there was any functional consequences. So I'm just going to show you some electrophysiological data. All we're doing is looking at the amplitude and also the frequency of inhibitory postsynaptic events here. So just the amplitude of these inward deflections, which are a readout for the strength of the synapse. So that essentially could be a readout for a change in GABA receptor activity, or potentially a change in the number of postsynaptic receptors. And so in collaboration with Eunice Yen and Zhen Yan at State University of New York, we looked at what happens if you incubate slices in the presence of this leupeptin inhibitor of the lysosomal degradation in terms of inhibitory synaptic transmission. And what they found was that there was a significant increase in the amplitude and also the frequency of spontaneous inhibitory postsynaptic currents. And actually, I mean, the easiest way to see this is really in the comparison of a control trace here or a leupeptin treated trace here. So the neurons there are far more events, and they're also mostly of a larger amplitude. So that is in agreement with the data that we have showing that the GABA receptor clusters are bigger, and also that there are more of them. So this suggests then that this degradation pathway is important for regulating postsynaptic receptor number and the strength of inhibitory synapses. But we were also interested to know what the molecular mechanism might be that actually shunts the receptors to this pathway. Because actually under most sort of resting conditions, for example, most of the receptors aren't targeted for degradation. They're usually rapidly recycled. And that implies then that there must be some sort of molecular switch that can basically decide whether or not this receptor will actually then then be targeted for degradation. And we thought that one way to figure out what the switch was to figure out whether or not within the GABA A receptor subunits there was some sort of protein targeting domain that might facilitate the delivery of the GABA receptors from this storage compartment here to the lysosome. And so in data that I don't have time to show you, Lorena carried out sort of extensive structure-function experiments to map which subunit of the GABA receptor might contain a lysosomal targeting domain, and also what sequence was mediating that targeting. And she found that there was a 15 amino acid or 15 or 20 amino acid region within the gamma-2 subunit of the GABA receptor that was able to facilitate this sorting of the receptors for degradation. So is there anything within this region that might actually be indicative of a switch that could occur to facilitate this lysosomal targeting? So what we noticed was that there were a large number of these lysine residues. So there was a lysine stretch within this domain. Now, lysine residues can be post-translationally modified. They can be modified with a small polypeptide called ubiquitin. And it's been known for a while that targeting ubiquitin molecules to lysine residues within membrane proteins can act as a mechanism for sending them to the lysosome for destruction. So we hypothesized that maybe, in fact, this region within the GABA receptor might actually be a target for this modification, and that could be important in some way for regulating receptor degradation levels. So to look at whether or not indeed that sequence was important, we carried out some fixed cell immunofluorescence confocal microscopy experiments where we looked at the distribution of internalized GABA A receptors, so the receptors that had been downmodulated from the plasma membrane, in different compartments within the cell. And here we're using a marker of the late endosomal and lysosomal system tagged with GFP called GFP-RAB7. So GFP-RAB7 is the green label in both these confocal micrographs. And then Lorena compared the distribution of GABA receptors containing alpha, beta, and gamma-2 subunits that had been downmodulated from the plasma membrane, and whether or not they co-localized with this GFP-RAB7 label in wild-type receptors, and also in receptors that had been mutated for this stretch of lysine residues. And what we found was that compared to the wild-type, there was much less overlap in these cells of the internalized receptors when we mutated out that targeting sequence, which is what you'd expect if that targeting sequence at the level of those lysine residues was actually mediating this relocalization of the receptors. And this is just some of the pooled quantified data. So is it also a substrate for ubiquitination, this region? Well, we can look at the ubiquitination levels of the receptor by expressing it in cells and co-expressing it with a tagged version of ubiquitin. It's then possible to affinity purify the receptors using antibodies and then run those affinity purified complexes on gels. So if the receptor is having this ubiquitin molecule attached to it via a post-translational modification to the lysine residues, then we can bring down higher levels of the ubiquitinated GABA receptor. And so if we run these complexes on gels, what we find is that the ubiquitinated receptor runs as a high molecular weight smear here. And in contrast, in the case of receptors that have this potential ubiquitination site mutated, we no longer see this high molecular weight smear. But we're still bringing down the same level of GABA A receptors. So that suggests then that indeed that region is a substrate for modification by ubiquitin. And so that allows us to propose a model whereby ubiquitination of the receptor acts as a signal to send the GABA A receptors to the lysosome where they can be degraded. So we were interested to know actually whether or not this might have some implications in terms of pathological changes in the levels of surface and synaptic GABA receptors. And the reason for this is that in a number of situations, such as for example in ischemia and also in epilepsy, receptors have been found to be rapidly internalized into an internal compartment. Now the stability of the receptors here, one could propose, would have important implications in terms of what happens to the strength of inhibition. Because if they're maintained here in a stable format, then they're available to be rapidly reinserted back to the plasma membrane and to repopulate these synapses. In contrast, if the receptors were targeted to degradation once they'd been internalized, for example, in ischemia or in epilepsy, then they're no longer going to be available to rescue this compromised synaptic inhibition, which could lead to a much more prolonged effect on the strength of synapses. So we looked basically at whether or not there was any effect on the stability of synaptic GABA receptors when we simulated ischemia, which Yuqiang Wang's lab had previously shown causes a downmodulation of receptors from the plasma membrane. And on the left here, these are experiments carried out by Lorena in collaboration with James Muir. What they did was to image GABA receptor clusters out in the dendrites. So each arrow here is a receptor cluster. And this is over a time where we simulated ischemia with oxygen glucose deprivation. And we found what Yuqiang Wang had reported previously, which is a loss of surface and synaptic receptors. So this is just the quantified change in fluorescence of the clusters over time. If we did the same experiment with the mutant version of the receptor that can't be ubiquitinated and targeted for degradation in the lysosome, we no longer saw this loss of receptors from the clusters. So this supports the idea that those receptors, they're being internalized and stored in an internal compartment, but then they can rapidly recycle to replenish the inhibitory postsynaptic domains. And so what that suggests then is that one way to be able to rescue a prolonged inhibition that might occur, for example, in ischemia where it's thought that ubiquitination levels generally rise, would be to, for example, target and block in some way the targeting of the receptors into this degradative pathway so that they could more rapidly be recycled back up to the plasma membrane to maintain those synaptic receptors. Okay, so in the last part of my talk, I want to focus on the mechanisms that are important for delivering receptors from this internal compartment back to the plasma membrane. And this is work carried out primarily by Alison Twelvetrees in the lab. And she wanted to know, basically, we know that there are these receptors that are contained here. We know that they can be rapidly sent back to the synapse and that can change the strength of the synapse. But how that process occurs had remained elusive for a long time. Now, as I said at the start of the talk, neurons are pretty complicated cells, and they have to transport things to many locations, and much of that transport is mediated along these microtubules, which basically serve as motorways for different types of motor protein to walk along these microtubule tracts. So one of the main families of motors that mediate this are called kinesin motors, and they use the hydrolysis of ATP in these motor domains here to basically provide energy to walk along the microtubule while this guy looks like he's working really hard, but the idea is that they're easily moving this vesicle along the microtubule motor. And so we wanted to know whether vesicles containing GABA receptors were targeted to synapses by a kinesin-type motor protein. Now, one of the problems, though, is that there are about 45 different kinesin motors. And so is there a way that we could sort of hone in or zoom in on one particular motor protein family? Well, there was some information that helped us in this because work that I carried out previously while working with Steve Moss showed that a protein called HAP1 can interact directly with GABA-A receptor intracellular domains, and that this HAP1 protein facilitates the reinsertion of GABA receptors back to synapses. But the mechanism of how HAP1 was doing this was unknown. So what do we know about HAP1? Well, HAP1 is a protein that was originally identified to interact with Huntington, which is the protein that gets mutated in Huntington's disease. And I'll come back to that at the very end of my talk. But HAP1 is also known to be highly expressed in brain. It was implicated in neuronal trafficking, and it had been shown or there was evidence pointing at the idea that it might be able to interact directly with some motor proteins. And specifically, there was evidence to support the idea that it could interact with a type of kinesin motor called KIF5. And that's a classical kinesin mediated by, made up of the homodimerization of two heavy chains. There are also these two light chains. The N-terminus contains the motor domain. The C-terminus is thought to be important for cargo binding. So based on this, we hypothesized that maybe the motor that's important for delivering GABA receptors to synapses was actually KIF-5. But to test this, Allison took advantage of a function-blocking antibody that can disrupt the function of this motor protein. So the antibody binds to the motor domain, probably locks it into some conformation where it can no longer walk along microtubules. So we used this antibody to test the hypothesis that maybe KIF-5 was important for regulating inhibitory synaptic transmission by regulating GABA receptor stability at synapses. So one way that we looked at this was Allison developed a system where she could couple the antibody to a membrane-permeant carrier peptide. And that carrier peptide drags the antibody into neurons, and so that allows you to transduce the neurons with the function-blocking antibody. We can then compare the effects of the SUC4, which is the name for this KIF-5 function-blocking antibody, compared to a control antibody that doesn't influence the kinesin motors. And then using immunofluorescence to look at the size of GABA receptor clusters out in dendrites opposed to inhibitory presynaptic terminals. So gamma-2 is the antibody that targets the gamma-2 subunit of the GABA receptor. VAT is an inhibitory presynaptic marker. Allison quantified the size of the clusters and found that blocking the function of the antibody reduced the size of GABA receptor clusters, suggesting that there were less receptors postsynaptically without the function of this motor. Another thing we could do is to introduce via the patch pipette the function-blocking antibody and then look simultaneously while recording what happens to the strength of inhibitory transmission. So here is just recordings from a neuron that's been dialyzed with the SUC4 antibody or with a control antibody. We saw this rapid time-dependent decrease in the amplitude of the inhibitory synaptic current, suggesting that there's a functional correlate to this loss of postsynaptic receptors. So if KIF-5 is indeed important for regulating the transport of GABA receptors to synapses, then one might expect to see the motor at the synapse, or at least in the vicinity of the synapse. So we tested this in two different ways. Firstly, Allison carried out some immunofluorescence experiments where again she labeled up GABA receptor clusters in the neuronal dendrites, here with the gamma-2 antibody. And then she also labeled up with antibodies to two different isoforms of KIF-5, KIF-5B or KIF-5C. And what she found was that there was some overlap, which you can see as yellow here, of the KIF-5 motor at some of these inhibitory postsynaptic clusters. So for example there. To get a better resolution of this co-localization, we teamed up with Antoine Triller and Philippe Rostin at the École Normale in Paris, who used an EM approach with KIF-5 specific antibodies to look at whether or not there were any of the motor proteins at or in the vicinity of inhibitory synapses, which were either identified morphologically or using stains for the for GAD in the presynaptic terminals. And what we found was that there was significant accumulations of KIF-5 nearby to these inhibitory synapses. So this is a zoom in here, and some of that KIF-5 looks like it's localized to some sort of subsynaptic membrane compartment, which might be a pool of receptors that are then being delivered back up to the synapse. So KIF-5 is in the right place. Is it actually in a complex with this HAP1 protein, which we were proposing was an adapter to link KIF-5 to the motors and also to link the motors to the GABA receptors? So to address this, we used a biochemical approach where we can immunopurify from, for example, a rodent brain, GABA receptors or other components of this putative complex, purify those out on beads, then run those on gels, and then look at what else comes down in the complex using Western blotting. So if we bring down the GABA receptor, we can bring down this HAP1, which are these two bands here. And if we bring down the motor protein, we can also bring down HAP1. And if we bring down HAP1, we can bring down KIF-5. And if we bring down the GABA receptor, we can also bring down KIF-5. So that supports then the idea that in native systems there are indeed complexes of the receptor in along with HAP1, which we think is the adapter, and the motor proteins. And so that allows us to propose a mechanism whereby GABA receptors are linked to these motors via HAP1, and that's important for the transport of GABA receptor vesicles. To further test this, we used another approach, which was a dominant negative approach, where we found a region that we could express at high level in neurons in the motor domain, the non-motor domain of the KIF5, and expressing that at high levels disrupted the complex. And then when we expressed that in the neurons, we looked at what that did to the surface GABA A receptor levels. And we can do this by labeling up the surface population with a biotin tag. That only labels the surface receptors. We can then purify out those tagged receptors on beads, run them on gels, and here we have the total receptor population in the neurons. And this is neurons that have been transfected with a control GFP protein or with a GFP tagged version of this dominant negative that disrupts the complex. There's no effect on total protein, but there's a significant reduction in the surface levels of the GABA A receptor, which is quantified here. So that suggests then that this is further evidence to support the idea that this complex is indeed important for regulating surface levels of receptors in neurons. Does that also influence the extent of synaptic inhibition? Well, when we express this dominant negative in the neurons, again we see, compared to control here, a significant attenuation in the amplitude of the inhibitory synaptic currents, which supports the idea that there may be a loss of synaptic receptors when we disrupt this motor protein-dependent trafficking. In agreement with that, when Alison did some imaging experiments where she looked at the size of GABA receptor clusters in neurons transfected with this GFP protein or with the dominant negative, again she found a decrease in the overall size of the GABA receptor clusters out in the dendrites, which is quantified there. So that supports then a model whereby HAP1 acts to link GABA receptor containing transport vesicles to motors for their rapid delivery to the inhibitory postsynaptic domain. And we think it's rapid because we can see an attenuation of the strength of the synapse within a few minutes of putting in an antibody that targets the function of this motor. So in the last couple of slides, I just want to cover a potential implication of this, the identification of this motor protein complex in a pathological situation, which is in Huntington's disease. So Huntington's disease is an autosomal dominant neurodegenerative disorder that's caused by an expanded polyglutamine tract in the protein huntingtin. And it leads to a number of things, including uncontrolled movements, psychiatric disorders, personality changes, cognitive decline, and eventually death within about 15 to 20 years of diagnosis. And the protein huntingtin is ubiquitously expressed in the brain and in other parts of the organism, but it seems to cause a selective neurodegeneration of specific neuronal populations, in particular the striatum and also the cortex. Now, it's thought that Huntington, in its mutant form, when you have an expanded glutamine repeat, so anything above 36 glutamines in the N-terminus of Huntington starts to cause disease pathology. So the mutant Huntington is thought to cause either a toxic gain or loss of function. Now, interestingly, HAP1 has been found to, as I said, interact with Huntington, and it actually interacts more tightly with the mutant version than the wild type. And there's growing evidence to suggest that mutant Huntington disrupts the function of this HAP1 protein in terms of intracellular transport in neurons. So we were intrigued at the idea that maybe mutant Huntington could actually disrupt the trafficking of these GABA receptor transport vesicles mediated by the HAP1 complex. To look at this, Alison did some cell biological experiments where she imaged the transport of these GABA receptor containing vesicles. So these are vesicles of GABA receptors labeled with GFP. And she did this imaging in a striatally derived cell line from either a wild type mouse or a mouse that has been engineered to contain an expansion in this polyglutamine repeat within the Huntington protein. And then looked at what happens to the kinetics of the transport of these vesicles. She found there was a small decrease in the velocity of transport of the vesicles and also a significant decrease in the run length of each vesicle. And this is over quite a short period of time. So this suggests that disrupting the trafficking of these, the mutant Huntington protein is likely to impair the delivery of these GABA receptor containing transport vesicles mediated by the HAP1 KIF5 complex. And if that was the case, one might expect to see less GABA receptors at synapses in neurons expressing a mutant version of Huntington compared to a wild-type version. And when we carried out immunofluorescence experiments to look at the size of GABA receptor clusters, indeed we found there was a depletion in the number of postsynaptic receptors that were opposed to the inhibitory labeled terminals with this marker VAT. So this is just the quantified data here. and that correlated with a decrease in the amplitude of inhibitory postsynaptic currents in neurons expressing the mutant version of Huntington compared to the wild type. So that suggests then that when we have mutant Huntington in the system, which is the red thing here, that disrupts the ability of the motors to be able to replenish the GABA receptors back to the surface and to inhibitory synaptic sites, and that leads to a slow but steady depletion of GABA receptors from this inhibitory domain, which is going to cause a compromised inhibition. Now, some other very nice work from Lynn Raymond's lab has also shown that mutant Huntington can actually cause an increase in the number of NMDA receptors being targeted to the cell surface. So conversely to what happens at inhibitory synapses, which basically get compromised, you get more activity in terms of glutamatergic signaling due to this mutant Huntington protein. So that suggests then that there might be sort of a double hit effect here, that basically mutant Huntington will compromise inhibition, it will enhance excitation, that could cause a disruption of the correct excitatory inhibitory balance, and that might lead to significant increases in calcium entry through NMDA receptors, which eventually could be quite neurotoxic to the cell. And so we would propose that that might be one contributing factor during the progression of the disease leading to neuronal cell loss. So I just want to quickly summarize what I've talked to you about today. I initially talked about the stability of GABA receptors at the plasma membrane and proposed that neuronal activity driven by the activation of glutamate receptors can change the phosphorylation state of receptors here, and that changes their confinement at the inhibitory post-synapse, causing them to move out of the synapse by lateral diffusion. I also told you that once the GABA receptors are internalized, we found that their decision as to whether or not they're targeted for degradation or whether they're recycled is in part mediated by whether or not the receptors get ubiquitinated, and that this decision can be important for regulating the number of postsynaptic receptors and might be particularly important under pathological conditions, which seems to drive receptors for degradation in this compartment. And finally, I showed you some evidence to support the idea that there's a motor protein complex between a motor called KIF-5, an adapter protein called HAP1, and the GABA receptor that's important for mediating the delivery of these receptors back up to the cell surface and to synaptic sites, and that protein complex may be a target for disrupted inhibition in a pathological situation in Huntington's disease, where mutant polyglutamine repeat-containing Huntington disrupts this trafficking by targeting the function of the HAP1 KIF-5 complex. Okay, so I just want to quickly acknowledge the people that did the work. So I've been very privileged since I set up my lab about 5 years ago to have a really wonderful set of people work with me. The initial imaging data looking at the lateral mobility of receptors at the surface was primarily carried out by James Muir, but also with some help from Andrew McCaskill and Kate Smith. Lorena Arancibia-Carcamo did the experiments looking at the targeting of receptors for degradation, and Alison Twelvetrees characterized this motor protein complex that delivers the receptors to synapses. We've collaborated extensively with Lewis Griffin at UCL, who's in computer science and who worked with James to set up the imaging and analysis programs that are necessary to be able to track quantum dots through time and reconstruct the trajectories, which is what we need to do to be able to get out important information about the receptor. And we've also carried out a number of collaborations with Zhenyan and Eunice Yen, who we worked with to, for example, characterize this motor protein dependent delivery of the receptors to synapses, in addition to with Antoine Trillo and Philippe Rostin. And Marcy McDonald and Frederick Sotou gave us some useful Huntington reagents. So thanks very much.